So let's pick up from our last class and continue our discussion on how to successfully cultivate microorganisms in the laboratory environment. Again, it can be a very difficult proposition due to the fact that we need to know, or at least try to predict, the types of nutritional and environmental factors that will influence the growth of a particular microbe. So let's now talk about media, something we've sort of alluded to previously in this chapter. Let's talk about media and the different types of media that are available. And based upon how they're classified, these different media types can provide you with varying degrees of information about the microbe or microbes that are growing on top of it or within it. So why do we care in the first place? Again, we need to think about that as well. And in microbiology, one of the most important roles of a microbiologist when trying to study a particular organism is trying to separate that species from the rest of the microbial community. And the way in which we try to do that, or the way we talk about it in the lab, is trying to isolate what's called a pure culture. So a pure culture is defined as a population of cells that are derived from a single cell. And again, referring back to the beginning of this chapter, we know that microbes, or specifically prokaryotes, all reproduce asexually. So as a result of that, when we place one cell on top of a plate or one cell within a tube full of broth, due to the fact that one cell is going to reproduce asexually and then generations are going to occur again and again and again and again, we're going to assume that if that, again, one cell was placed in that one particular medium, that all of the cells, all of the offspring that result later on in the future are going to be genetically identical. So we place one cell of E. coli on top of a plate. We're going to assume that when we look at that plate 48 hours later and we see this large colony of 1 million or 1 billion cells, they should be all clones of each other. And the way we do that are ways that Robert Koch developed in the 19th century, which are aseptic techniques. And if you're in the lab right now, you're going to be covering these if you haven't done so already. So I won't go into that. But one of the ways we try to do that is going to be, if I go through here to this slide, is what's known as the streak plate method, which is basically a way in which we try to alleviate the problem that it's hard to isolate one particular cell of a microbe on top of a plate. Because we're dealing with cultures and we're trying to move typically, or we're trying to transfer a tube that's filled with millions if not billions of cells, how do we take a small enough fraction of that liquid and transfer it to a plate so that we can actually get individual cells separated from each other? If we can't do that, then we can't assume that we've attained a pure culture, because all of these different cells are going to be mixing with each other. The colonies are going to melt together as they grow, and you're just going to see basically a big smear, which is sort of exemplified here on this portion of the plate. So we use a technique like streak plate method, and you can read about it in your book, and if you're in the lab, you'll cover this if you haven't done so already. But this is a way in which we can, over time, or over the course of this plate, get to a point in which we're separating the cells enough, we're diluting the population enough, in which we can assume, or we can actually observe, separated colonies, which then we can assume are all genetically identical.